Jake Godfrey. He will be talking about aggressive females, my counteract male pace of life trade-offs. And I see the screens coming. And yeah, feel free to start when you're ready.
Excellent. Thank you very much. Do you hear me loud and clear? Yes.
Yes.
Perfect. Thank you. Just like to check. Thanks everybody for kind of hanging out and listening to my talk. I really appreciate it. Hopefully I won't make you regret it too much. I'm going to be talking to you today about basically how some aggressive females might effectively be or have led to some counteraction of some pace of life trade-offs. In this work, I'm really excited to present it because it's really new. This is a really new data set that I've just got kind of a quick run through of analysis on. And this is all branching off of some previous work I've done on this species right here, Tigrosa heluo. And basically, I've been doing a lot of investigation of the pace of life syndrome hypothesis using them as a kind of a model organism. So the first thing I want to do is kind of just go over what is the pace of life. What are we talking about when I'm talking about this? Where did this idea really come from? And this is really just a broad idea that organisms can develop along this slow, fast continuum. Some organisms or some species or some individuals might mature more slowly, delaying their reproductive opportunities, while they can possess longer lifespans. Perhaps instead they're investing in long-term survival traits. While on the other end of the spectrum, you have those that might mature really, really rapidly, investing heavily in this development right up front to try to maximize or get to those early reproductive opportunities. And this was really kind of brought into the idea of this pace of life syndrome hypothesis, where we are trying to kind of explain a lot of covariation and the pace of life observed, as well as various phenotypic traits of organisms. And there have been a lot of studies looking at this and looking at a lot of different traits, trying to see if there are perhaps common selection pressures that really kind of put pressure on different traits to comprise a complete syndrome. Just to give you an example, and these are kind of 5 traits that I've investigated with this species of Tigrosa, and it's really kind of driven me to ask this question I'm about to ask. We looked at adult body size. It's been shown in a lot of different species that the faster I develop to maturation, the smaller I will be as an adult. There's also a lot of cool studies on pigmentation or coloration, and basically how it can correlate with metabolism, which is really, really, really heavily correlated with pace of life. And so we see those on different trajectories, basically having different pigmentations or colorations. Also some behaviors. As a subadult, I might have a little bit more aggressive behavior if I'm trying to mature more rapidly, because I might need more resources to invest in my maturation. Asymmetry, there's been a lot of cool studies looking at how ramping up or speeding up my development time might lead to more asymmetrical features in my adulthood, which can have play a really, really big role in mating success, for example. And finally, the one that I'm really going to talk a lot about today is immunocompetence. And there's been a lot of studies on a lot of different species of birds, for example, as well as looking at some amphibians and reptiles. But basically, if I am investing heavily in maturing and getting to a reproductive stage quickly, I'm not investing as much resources in my immunocompetence. I won't be able to deal with invaders as effectively. And so I mentioned that I've been looking at this species of Tigrosa, and I've really been trying to kind of evaluate them for what kind of constitutes their pace of life syndrome, what all correlates with their pace of life. And in a previous study, I found that basically slow maturing individuals are larger and heavier as adults. That's really, really lining up with the literature that's already established. I've also found some really cool results with their coloration. So I have photographed all of their carapaces with those that I reared from egg to adult. And I was able to map on, or basically correlate, their coloration or the contrast of their carapace with their development speed. Also, their behaviors. Now, I looked at their behaviors as adults, and it was actually counterintuitive. It was opposite of what we were expecting. These slow maturing individuals were actually bolder as adults, and this might be due to their just larger body size. They're a little bit less frightened or a little less skittish. They can kind of have their way around the novel environment if they want. But two things kind of came back not necessarily linked with pace of life, and that's asymmetry, which we saw, or which we have seen in other species is linked to pace of life. It wasn't linked in this species, as well as encapsulation ability, which is really my measurement of immunocompetence. And I'll talk about that in a couple of slides. But I did not find any correlation with development speed and their encapsulation ability or their immunocompetence. But one really interesting thing that kind of stuck out and really brought me up to this question or this hypothesis that I've kind of posed for this talk is that we saw greater encapsulation rates for males. These really, really short-lived, shorter lifespan males comparatively to the females of the species, they seem to be investing more in their immunocompetence. They seem to deal better with foreign invaders in their body than the females actually do, which really just kind of threw me through a loop. It didn't make sense as far as the pace of life syndrome hypothesis is concerned, and it's something that I wanted to investigate. So, why is this happening? And we kind of came up with this hypothesis. Perhaps we're getting a lot of pressure from females on the males, and that's really driving up their immunocompetence. We know in this species that the risk of sexual cannibalization is really, really high. More than 30% of mating trials in a lab will end up with sexual cannibalization before mating can actually occur. So it's really, really high risk. And we also know from other studies that females might actually favor males with higher immunocompetence. We know from honest signals such as four-leg tuft size and schizocosa, or even male courtship vigor in several different species. This might actually be an honest indicator of basically the male's immunocompetence, and it's actually favored by the females. So that's kind of led us to a question of or a hypothesis that female selection pressure during mating is actually kind of pushing or counteracting what we would have normally seen with the pace of life syndrome. So in order to test this, we completed a lot of mating trials. There was 56 of them in total, where we basically allowed adult virgin tigrosa to, we gave them the opportunity to mate. Now, in order to take immunocompetence, in order to perform these encapsulation assays, which you'll see on the next slide, this is a destructive process. So I needed the males to survive the trials, the mating trials. So unfortunately for this, I had to superglue the female mouth parts in order to prevent any actual destructive or cannibalization of the males. With that being said, I noticed that the female's behavior stayed the same. She would aggressively lunge at males that she was not interested in, or she actually displayed a receptive posture for those that she was. So it seems like they were still kind of getting their point across just without the cannibalization, so that I could kind of use the males later on. So I measured multiple different things that are commonly measured on males in order to kind of correlate or kind of figure out what determines mating success. And I looked at immunocompetence, and to do this, I used encapsulation assays. So what you see here are 3 different nylon filaments, each of 2 millimeters in length. And what we do is we implant these surgically into the male in his abdomen, and we basically allow that filament to stay in place for 24 hours. After which we freeze the male in order to halt the encapsulation process, and we can then look at these, photograph them, and quantify how much melanin was actually placed or encapsulated on these filaments. The darker these filaments were, like the one at the top, means more melanin was kind of surrounding that foreign object of the body, and it was more secluded. So this is dealing with that foreign invader. You can imagine something like a nematode, trying to kind of seclude that and get it away from the rest of your body so that it can't do any more damage. While those that might not have as high immunocompetence, they're not great at encapsulating that thread. It gets a little bit lighter, and that way we can compare these and make comparisons. I also, of course, used body condition measurements because we wanted to see what's really driving this. We wanted to kind of bring in all of the normal kind of control variables as well. So we took basically the reciprocals of mass over carapace width for all of these males in order to get a measurement of body condition. I also wanted to look at asymmetry. This is something that's in the literature as well as having a big impact on mating success. So what I did is I took the tibia patella lengths, thank you, of both forelegs, and I took the absolute value of the difference between the two in order to get just a metric of asymmetry for comparison. And finally, so something I've been including in all the studies that I've been doing is just this measure of coloration or contrast. Basically, how bright or light are these individuals? So I would photograph all of the adult male carapaces and basically use image J to get an average or a mean contrast or brightness of their carapace, just to see if that might be a cue to the females for how these males are. So what did we find? Well, pretty much asymmetry runs the board. Asymmetrical males, the more asymmetrical I was, the more likely I was to be met with aggression from a female. So here on the x-axis of this figure, you've got whether or not a female was aggressive towards the male at all. And on the y-axis, you've got the average asymmetry value of that group. So here you can see the group that was more asymmetrical, they were the ones more likely to see aggression. We also saw that not only did they see the initial aggression, but they also saw more attacks. So the number of females' aggressive lunges that were shown towards the male, this increased with the more asymmetrical the spiders were, the males were. But on a really cool kind of topic, or what we were kind of looking for, is we also found that even more strongly correlated was that immunocompetence. The lower my encapsulation score, the more aggressive a female was. So here on the x-axis, you've got the male encapsulation, while on the y-axis, you've got the number of female lunges. The higher the encapsulation value is basically the better I did with that foreign invader, the more I was able to securely basically partition that invader off with melanin. So really, that was what we were looking for and what we were really most excited about. And then, of course, just asymmetry in general, really predicting how successful a male is at mating. The more asymmetrical I am, the less likely I am to actually successfully mate with a female. So this kind of short spin-off from my pace of life syndrome investigation and kind of trying to just look at some different outcomes that we saw and trying to make new hypotheses, it really came up with some cool conclusions. So female aggression may have actually played a role in driving the increased male immunocompetence. If I am being attacked more frequently by females, I'm probably going to not survive in order to go mate with another female, even though I was rejected previously. So it really could be driving kind of this shift from the pace of life syndrome that we would be expecting. And it's also really interesting to think about how sexual selection or other pressures might actually be so intense that the pace of life trade-offs just become blurred. And it really kind of presses the need to understand the species that you're working with and all of the different pressures that are on them. And you need to understand that before you attempt to apply a lot of the pace of life syndrome assumptions or ideas. And finally, it was just really cool to use this pace of life syndrome hypothesis as a tool to kind of develop or test, in my case, a new hypothesis, but you could do this for a number of different hypotheses, just based on divergences of what you expect and what you've actually observed. I found it was very, very interesting, led to some really neat results that we wouldn't have looked into without. With that, I don't want to take up any more of your time. I want to thank you all for listening to me. And of course, I want to thank the American Arachnological Society as well as Miami University. Without either of those, this work would not have happened. So I really, really appreciate it, as well as everybody else that helped me with these studies. Thank you very, very much.
Thank you. It was a very interesting talk and a very nice experiment. And unfortunately, we don't have time for questions, but I guess people can approach you with questions afterwards if needed. And
Definitely.
yeah, and with that, I would like to thank for all the amazing talks and for everyone who attended this
Gustavo,
behavior
I'm sorry, I don't
session.
want to. I'm just gonna, Jake, you can do a couple questions just because we're at the very end, and I know that you're in the student competition. So maybe
Oh,
just take
okay.
one or two
All right.
really quickly for him.
Okay. Yes, for sure. Thanks, Sarah, for bringing that up.
Jake, are you still there?
I am.
Okay. Okay, I'm going through the chat here. There's okay. The question just came up here in the chat. Do you think male's tendency to wander further than female to find potential mate affects the data? Yahoo Kim asked here, I believe as they travel further, they may be more exposed to antigen and parasite. Does evolutionary need more immunocompetence?
Definitely, and this is a really cool point and something else that I haven't really looked at or messed around with, but it's been in the back of my head, is the idea that the males are the ones that are typically more active in searching out females as far as mating is concerned. So they're actually having to travel further. They are more likely to come in contact with parasites or bacteria, things that they need to actually seclude from the body. So in that case, that could actually be also contributing to this divergence that we're seeing, where the males are actually having this higher immunocompetence than the females. It's definitely something that's really, really cool to think about, and it's probably, of course, like everything in biology, it's something that's happening in concert, where you have all of these different factors putting all these different pressures. But I think that that's a very key one that you mentioned. Yes, definitely.
One more came in here in the chat. If you want to address, is there any statistical interaction between color contrast and immunocompetence?
Yeah, I didn't. So I didn't include any interaction effects because there weren't any. In the previous study that I did, I actually investigated these a lot more. I raised them from eggs to all the way to adult and looked at if I could correlate anything with color and immunocompetence, because that's what I was hoping for, melanization with coloration kind of a thing. But there weren't actually. That's not correlated at all for this species, which was kind of surprising to me and not what I was expecting.
Yep. Yeah, I thought this asymmetry was very interesting. Also, and Mariella asks here if you were able to test or if you plan to test the effect of male asymmetry on reproductive success.
Yeah, so I wasn't able to test that, but I think that that's something that's extremely easily testable and an obvious next step. And it's something that I have an undergrad working with me next year. I think that that might be a good project to work for, and it's something that is still to come, definitely.
Okay, there's one more from Luis. Luis Garcia, do you know how frequent are these asymmetrical males on natural populations?
I do, but I don't know off the top of my head. So one of the studies I did, I actually did just a survey on adult males or on adults in general, adult male and female. So I have all of that data. Unfortunately, that was 3 or 4 years ago, and I cannot recall now to save my life. I apologize. I can get that to you though. But I do have all the data on asymmetry on individuals in the wild as well. I just can't recall.
It's fine. No, very cool. Thanks for the answers. Thanks for the discussion.
Thanks, everybody.
Thanks everyone. Yeah, thanks everyone for coming. So see you around in other talks.